Hi all, I would like to welcome you to today's seminar organized by Hellenic Initiative of Against Alzheimer's Disease, HIAAD. For those who are new in this seminar series and don't know lots about the HIAAD, I would like to say that it's an initiative started more than 2 years ago with a major goal to implement a Greek National Dementia Action Plan to fight Alzheimer's disease using a range of approaches, one of which is to promote the research in the Alzheimer's disease field. So HIAAD, it's an initiative coordinated by the Precision Medicine Center of Excellence for AD at Johns Hopkins University in the United States and the Bioinformatics and Human Electrophysiology Lab in Onion University. Our central objective is to promote the basic science in dementia in Greece by stimulating and expanding research collaborations. And we hope to achieve this in part by organizing this monthly seminar series with invited speakers from all over the world to exchange ideas and promote discussions and collaborations on AD research. Today, we have an exceptional speaker, Dr. Shane Liddelow, and Dr. Rebecca Matsas is going to introduce you. Thank you very much for attending the seminar. Rebecca?
Thank you, Vaso. It is a real pleasure today to have with us Professor Shane Liddelow from my New York University Grossman School of Medicine. Professor Shane is a real expert and has pioneering contributions in astrocyte research. He obtained his bachelor's degree and his PhD from the University of Melbourne in Australia. I just found out that he comes from Croatia. So he's done a lot of traveling during his career and his life. As a postdoctoral fellow, he joined the lab of Ben Barres at Stanford University, a lab, again, with pioneering contributions in astrocyte research. And there, Dr. Liddelow discovered a subtype of reactive astrocytes that release a toxic factor that kills specific subtypes of neurons. And these type of astrocytes are present in the brains of patients with several neurodegenerative diseases. In 2018, just 4 years ago, he started his own research group at the Grossman School of Medicine in New York. And his research focuses on the multiple reactive responses of astrocytes to infection and disease. He has uncovered heterogeneous substates with altered function and has been developing tools to further research that could be also available in the community to uncover drug targets specific to individual reactive astrocytes' functional states. Professor Liddelow has been publishing in top-notch scientific journals, and he has received a number of awards. And today he's going to talk to us about what do reactive astrocytes really do. So, Shane, what do they really do?
Well, I have to say first, that was a really wonderful introduction, Rebecca. Thank you for that. And thank you also to the HIAAD for this really wonderful opportunity. To answer your question, though, I don't know. I don't know what reactive astrocytes really do. I think that's an incredibly complex question. And we know some things that they do, but we don't really know everything that they do. I don't think anybody does. But I hope that collectively as a field, we are slowly getting closer and closer to a more comprehensive understanding of all of the different sort of menagerie of wonderful tasks and functions that these astrocytes have. So what I want to tell you about today then is part of that answer that I think my lab is going to be chipping away at for a long time. And I'm going to take us on a little journey, sort of starting with some of the historical work that we have done and others in the field have done, move into some functional characterization, particularly as Rebecca mentioned, the identification of this neurotoxin that we published late last year in Nature. And then I want to move on back into Alzheimer's disease specifically and talk about some recent work, one that's available as a preprint, and I'll share those details with you at the end of the talk today, looking at Alzheimer's disease mouse models and astrocyte heterogeneity. And the other is actually a paper that came out in print yesterday in Neuron, looking at astrocyte heterogeneity in human Alzheimer's disease patients. But what I want to do, though, is actually want to start with the people who actually did the work. I am learning that I get to do less and less actual bench science nowadays as the lab grows bigger and bigger. So really, I think it's important that I highlight these exceptional individuals. And the work I'm going to talk most about today was spearheaded by 3 people in my lab: Michael O'Day, an amazing graduate student; Jessica Sadie, the postdoc who's now moved on to a new position; and Philip Hazel, another postdoc in the lab. But the vast majority of the biochemistry identification of the toxin that I'll talk about was actually done by an amazing graduate student, Kevin Gutenplan, who's at the top right here. And I was very fortunate to work with Kevin very closely when we were both in Ben's lab at Stanford. And all of these people were ably aided by everyone else on the slide and numerous others that I'll highlight with faces and names as we go through the talk. So what I want to start with, though, is really a description of what it is I think my lab does. You know, we ultimately want to answer this question: what is it that reactive astrocytes really do? But at its heart, we're cell biologists, and what we care about is how one cell in the central nervous system interacts with another cell in the central nervous system. And the dichotomy here is, how does that change under normal physiological conditions and under a whole suite of pathological conditions? And so for today, I'm going to focus on astrocytes, which is what most of what we do is focused on. But we're going to focus on astrocytes and their interaction with neurons. And if we think about the physiological role and we break this down and simplify it to a level that is way too simple, but we can think about a very basic function in that astrocytes provide support to neurons, neurotrophic support. They provide nutrients, they take away waste products. And that's a really important component of this interaction between these two cells. And ultimately, it's neurons that we want to keep alive because they fire action potentials and make us have memories and live and laugh and walk and do all those amazing things that as organisms we are able to do. Now, in the context of different pathologies, whether they be acute infections or inflammatory insults or chronic diseases like Alzheimer's disease, a whole host of things can happen to these astrocytes to drive them to a state that we would call broadly a reactive state or astrocyte reactivity. And if we simplify this, we can say, well, maybe these astrocytes become less able to support neurons. That is, the neurons could be functionally ineffective or could die. And we think about this occurring in a number of different ways. One is there could be a loss of function. So the astrocytes don't do what they do normally. So maybe they don't provide enough trophic support. Those neurons slowly become dystrophic and they would die due to a lack of nutrients. Or maybe, as we'll spend a lot of time talking about today, these astrocytes could gain a function they don't normally have. That is, this gain of function, and that would be that they would release a toxin that would target specific neuronal subtypes. But of course, it's not this simple. It's not just they move from being helpful to being harmful. It's very, very complex. And there are substates of reactive astrocytes that can have a gain of function that is actually highly protective of neurons and of other CNS cells. And we'll talk a little bit about this towards the end of the slide deck today. So how do we study this, right? So we're interested in astrocyte function. We're interested in reactive astrocyte function. And we know that there's a lot of heterogeneity in this response. And so how do we actually set about planning out these experiments and going from an idea through to actual function and ultimately targetable subset or druggable target on a subset of astrocytes? So the first thing that we do is we start with some transcriptomic data. It's very, very simple. It's cost effective. I won't say it's cheap, but it is cost effective to do. And you get an idea of genes that are differentially expressed with respect to a different pathogenic protein or disease, or you get an idea of the heterogeneity across different species or sexes or brain regions, etc. We then try and recapitulate that gene expression signature in a model that can be used to investigate the function of that subtype of cells. And historically, this has been done largely with cell culture experiments because you have a lot of control over actually studying individual cell functions. And you can piece together the individual cells of the CNS to see how they interact with, say, neurons, oligodendrocytes, microglia, etc. But every one of these in vitro studies has to be validated back in vivo to make sure that the assay that you run is not giving you an output that's an artifact of your culture system. And we've had a number of successes over the years of studying a number of different subtypes of these reactive astrocytes using these assays. And then, of course, once you've got that, you do the really complex thing, which is look at all the possible variables that could change this function. So it could be changing across age, maybe developmental astrocytes respond differently to that stimulus than senescent astrocytes. Maybe there's differences with respect to males and females, across different brain regions, across disease pathologies. Maybe what we see in a rodent doesn't happen in a human, so it could be species specific. There's a whole host of variables that we then have to validate to make sure that what we're studying is actually relevant for patients with, say, Alzheimer's disease. So what are some of these functions that we study? And I mentioned just interactions of neurons before, but very brief, I'm going to run over this again because I think this is really important. We sort of mentioned the whole idea of trophic or metabolic support. We know that astrocytes keep neurons alive. We also know that they're really important for secreting molecules that are required for synaptogenesis, things like glypicans and thrombospondins from Nicola Allen's lab, Charlotte Eroglu's lab, etc. We also know that astrocytes interact with the periphery, whether that be infiltrating immune cells, interacting with the blood-brain barrier, but they also interact with resident immune cells, microglia as well. And these are functions that are really important, and we start asking questions, do the astrocytes still have these functions? So, for instance, if there's a loss of synapse density, which occurs very early and prodromally in Alzheimer's disease, is that due to excess microglial pruning, which is what people like Su-Yon Hong at UCL or Beth Stevens has worked on, or is that due to a lack of formation of new synapses, which would involve astrocytes? We can also then look at, well, maybe astrocytes don't lose functions, they just become less effective at them. So maybe they don't stop providing trophic support, but maybe they just provide a little less of it. So the neurons are always running on empty, so to speak. They don't have enough nutrients to do their functions. Or maybe these astrocytes don't reuptake and recycle excitotoxic neurotransmitters like glutamate properly. And there's a whole host of literature, particularly in the ALS field, looking at astrocyte reuptake of glutamate and how this could be detrimental to neuronal health. And there's a lot of this actually happening at Hopkins, which is really exciting. Then, of course, there's the possibility that astrocytes gain new functions. And we already mentioned earlier that perhaps these astrocytes could re-enter the cell cycle, form a border or a scar around some necrotic tissue or some pathogenic protein and preserve neuronal health and function. Or what we'll talk about today is the fact that they could gain this ability to release a molecule that could be detrimental to neuronal health. So I sort of wanted to start this right from the earliest place that we started looking at this problem all the way through to identification of a neurotoxic compound. And I always like to start with this piece of work from Jennifer Zemanian. It was published exactly a decade ago now in the Journal of Neuroscience. And this is a very, very basic investigation of astrocyte heterogeneity or reactive astrocyte heterogeneity. And of course, as you'll see throughout the talk, we do this with far more exuberant techniques nowadays. But this is a really fundamental paper, I think. And what Jennifer did was took a astrocyte reporter mouse. She gave this mouse 2 insults, an acute inflammatory insult using lipopolysaccharide, a bacterial cell wall endotoxin, and she also gave it an ischemic insult where she ligated the middle cerebral artery. She saw they had very different transcriptomic profiles, suggesting that there were 2 reactive astrocyte responses. And this was only 2, right? It's very, very simple, very basic. But I think there's 2 really important sentences in the abstract of this paper that I think the field largely overlooks. And the first is that this raises the question of exactly how many subtypes of these reactive astrocytes there are, not stating that there's only 2, suggesting this is 2, but there's likely more. And I think we've found over the years that, of course, there are many more. And the other thing is that this is not the end, this is just the beginning. And what we do now with this transcriptomic database is we try and work out exactly what their functions are and testing them with hypothesis-driven research. And that's exactly what we've been doing ever since I read this paper of Jennifer's a decade or so ago. So a one-page summary of the work that I did while I was at Stanford is just to show that we did this looking at this neuroinflammatory subtype of reactive astrocytes. We worked out what drives their formation. It was immune cell or microglia release of cytokines, IL-1, TNF, and C1q. We knew that we could model this both in a dish and in vivo. We could drive them, we could stop them, we could look at what their ultimate function was interacting with all these cells. And importantly, we could also visualize them in human postmortem tissue, because at the time we actually did single cell PCR, which was not easy. nearly a decade ago, but we did do single cell PCR and saw that they upregulated specifically this complement component C3. So we had this correlative sense that they were in the regions of degeneration in brains of patients with, say, Alzheimer's disease or multiple sclerosis. But because we had a transcriptomic signature and a model, we could study their function. And so we asked very simple questions. Do they lose any normal functions? Do they gain any new functions? And would this enable us to have a better idea of exactly what these cells might be doing in the context of disease and pathology? So I won't go through all of the functions we studied, but a few of them are, if we go around this wheel, we know that these astrocytes fail to promote the formation of structural and functional synapses, which is really important. We already talked about the prodromal synapse loss in AD. We also know that these astrocytes completely downregulate their phagocytic pathways. The receptors like MERTK and MEGF10 that Wonsok Chung had looked at in mouse, or when this was done by Mark Freeman, it was Draper in the fly. But we saw that these receptors were downregulated, and we saw that they were no longer able to uptake cellular debris, myelin, pruin synapses, or uptake amyloid beta. And there's a whole host of other functions that they lost as well. And of course, important for this story, we found this one gain of function effect, and that is that they had this very potent capacity to drive the death of mature neurons and mature oligodendrocytes, but remarkably not any other cells in the CNS. All other cells, astrocytes, microglia, pericytes, endothelial cells, tanycytes, plexus epithelial cells, everything that we tried was also not able to be killed by whatever this toxin was. So there's two questions that came from this. First is, of course, what is the toxin? And we'll get to that in a few slides. But the other was, well, is this just an artifact of either culture or rodents? We validated all of these assays in vivo, so we knew it was not an artifact of culture. But there was always this niggling doubt that even though we had stained some postmortem tissue and correlatively shown that these astrocytes were present in regions of neurodegeneration, we didn't actually know if the drivers of this subtype were the same in human astrocytes and if their functional deficits were the same in human astrocytes. So when I moved to NYU, that's exactly what we decided to investigate. We did this with a very long-term collaborator and friend, Valentina Fossati, at the New York Stem Cell Foundation. And all this work was spearheaded by Lillian Babar, who was a technician in Valentina's lab, and Indigo Rose, who was a technician in my lab at the time. They've now both gone on to medical school and grad school. But what we did together was used human iPSCs to drive the differentiation into astrocytes, and then studied the function of physiologically normal astrocytes and neurotoxic or neuroinflammatory reactive astrocytes, using these same three mediators that we'd used in the rodent studies, IL-1, TNF, and C1q. And when we did this, we saw not only a recapitulation of the same transcriptomic signature, so at least that was evolutionarily conserved, but we also saw a conservation of the functional deficits as well. So failure to promote the formation of structural and functional synapses, failure to decrease phagocytosis or reuptake of glutamate, and the ability to drive the sort of formation of some sort of unknown toxin to drive the death of these neurons as well. So again, this was a subset of reactive astrocytes that was identified using transcriptomic studies. We worked out how to study them functionally. We validated it in vivo, validated it across species, and now we have this really strong feeling that this was a subtype that was really important to the pathology in a whole host of different neurodegenerative diseases. So just to sort of reiterate that a little bit, we know that microglia are able to drive this state. We know also that these neuroinflammatory reactive astrocytes can release a toxin, but at the time we didn't know what that was, and we'll go through that a little bit in the next few slides. We also know that we could actually block these astrocytes to preserve some neuronal health and function, and I'm going to give you a little vignette of that coming up next. But what we didn't know, because everything that we had done up until this stage was using acute insults and acute injuries. So maybe this was just a response that happened acutely, maybe if you have a traumatic injury, but it's not something that would persist in a chronic disease model or in patients with chronic neurodegenerative disease. So we actually decided to investigate this, and this is some work that was spearheaded by Kevin Gutenplan. It was published now 2 years ago in Nature Communications. And so we're going to jump out of the Alzheimer's space for a little bit here. And the reason for that was I was a new lab. I didn't want to have anyone working on a project that would take 18 months to age mice. And so we looked for a chronic neurodegenerative disease model that was a lot more aggressive. And the model that we chose was the ALS model, or the motor neuron disease model, with a mutation in the superoxide dismutase gene, this SOD1 G93A high copper mutant mouse. And the first experiment that Kevin did here was he took spinal cords from SOD1 mice and wild type or normal mice, and actually looked in the spinal cord to see if these neurotoxic astrocytes were even present. So he did some staining, or in situ, I should say, for C3, this marker of these neurotoxic astrocytes. And he saw in the wild type mouse, there was none of these astrocytes present, but in the mutant mouse, the SOD1 mouse, there's a large increase in the number of these astrocytes present. And then this is a comment for the trainees on the line. I then told Kevin not to do the experiment that he then did. And ultimately that experiment was to take advantage of the SOD mouse and to take advantage of this really dirty mouse model that we had used in earlier studies. And this was a global knockout for all three of the microglia-derived signals that were driving this neurotoxic phenotype. And I say it's a dirty model because it was not cell type specific. It was obviously not astrocyte specific at all. Those factors are really important for synaptic plasticity and development. And so ablating them from the entire animal is not the cleanest of models, but we had used it very effectively in acute injury paradigm. But Kevin didn't listen to me. So he crossed an IL-1 alpha knockout with a TNF alpha knockout, with a C1q knockout, with an astrocyte reporter, and then a SOD1 mutant mouse, and ultimately asked the question, if these astrocytes can't be formed, do the mice do better? And so the first question is, well, can they not be formed? And it turns out that that's true. If we knock out the inducing factors, there's a marked decrease in the number of these C3 positive astrocytes. And then the big question is, do they do better? And indeed they do. Here we have just a survival curve. In the vertical axis, we have the survival rate of these animals, or the viability of these animals. In the horizontal axis, we have the age of these mice in days. And we can see in red, the general SOD1 mouse is normally dying due to hind limb paralysis, difficulties breathing and eating within 3 to 4 months. But in the mice that no longer have these neurotoxic astrocytes, we see an increase in the lifespan of these animals of about 50%, which is a really remarkable increase for this rather aggressive neurodegenerative disease model. So this is all in mice. Of course, we're always interested in whether or not this is true in humans. And so the other thing that Kevin did was actually took advantage of some human postmortem tissue, went in and stained for GFAP to label astrocytes, or all astrocytes, C3 to label these neurotoxic astrocytes, and just asked, in what populations of patients are these cells even present? And the first thing that he did was look in non-symptomatic control patients. Obviously, there was astrocytes. There was no neurotoxic astrocytes. He then looked in patients that were harboring a mutation in the SOD1 gene. My computer froze, so hopefully you can still hear me. Here we go. And we can see that while there is still astrocytes present, there is now an increase in these C3 positive neurotoxic cells. But he also looked in patients that don't harbor a mutation in SOD1, so other mutations associated with the disease, like in C9orf72, and also in these sporadic or so-called non-genetically predisposed patients, he also saw a presence of these C3 positive astrocytes. And this didn't matter if he looked in the spinal cord, in the medulla, or in the motor cortex. In this graph here where he's quantified this, there was again a marked increase in the number of these C3 positive cells in all patients, regardless of their genetic predisposition. Now, of course, the caveat here is we only looked at a small number of patients. How indicative this is the entire population. So Kevin also took advantage of some whole genome sequencing through Project ALS, where they have hundreds of patients that have been sequenced. And he saw that in patients with ALS, compared to non-symptomatic controls, there was a marked increase in just the raw expression of this C3 marker. And again, if you split this out according to different diagnoses or different predispositions of these patients, regardless of whether or not they have mutations in SOD1, C9orf, other genes like TDP or FUS, or again, if they're pre-symptomatic, there was a marked increase compared to the non-symptomatic patients. So the very last thing that Kevin did then was then ask, well, is it the astrocytes? You know, a lot of historical literature from Kevin Egan's lab or from Serge Preborsky's lab had suggested that it didn't really matter what cell expressed this mutation. Ultimately, you would get the pathology so long as it was a non-neuronal cell. It seemed that microglia or oligodendrocytes or astrocytes would all drive this pathology. So what Kevin did then was just some very basic sequencing experiments. This is bulk sequencing from purified astrocytes on the top, purified microglia on the bottom, And in the first instance, we're comparing mutant cells versus wild type cells in a quiescent physiological setting. And what you'll see is, of course, these are volcano plots, I promise. It's just that there's no genes that are changing their expression levels. So this we interpreted as pre-symptomatically, both microglia and astrocytes were normal and were functioning normal and had normal gene expression signature. Kevin then did a very basic experiment that hundreds of labs around the world have done. This is nothing new. He took wild-type astrocytes in a quiescent and activated state, or microglia in a quiescent activated state. He saw thousands of alpha and downregulated genes. This was to be expected. But the really insightful experiment he then did was took the mutant activated cells and compared them to wild-type activated cells. When he did this, he saw that there was no difference in the response of the microglia, but there was a marked difference in the response of the mutant astrocytes. They responded with some different genes they didn't respond with previously. And even more surprising than that, and I apologize that this image is quite pixelated, but what we're looking at here is actually the changes of this maximal astrocyte response to changes in the stimulus input. So in black, we have all of our wild-type astrocytes. In red, we have all of our mutant astrocytes. And we can see when we give a full stimulus here, these astrocytes respond almost identically. But if we drop down to around 5% of this stimulus, the wild-type response has decreased to around 50%, whereas the mutant response has maintained at around 85 or 90%. And so the way that we interpreted this is not only were the astrocytic responses in mutant astrocytes increased, actually the threshold for that activation was markedly decreased so that we could actually stimulate them with around 5% of the same stimulus as a wild-type cell to mount almost a full response, which could be incredibly detrimental for patients that actually are initially triggered by these secondary inflammatory stimuli. But of course, the big lingering question here is not can we keep mice alive, not are the astrocytes responding differently, it's really what is the actual toxin that is released. And so what I'm going to do now is take us through a vignette of some hardcore biochemistry that runs through exactly how Kevin was able to identify this. So the first thing I want to show here is just a very simple experiment where these are actually retinal ganglion cells here in phase. And a number of years ago, we were adding toxic conditioned astrocyte media to these cells. And we saw that unlike removal of trophic support, which would take a week to 10 days for these neurons to die, the addition of this astrocyte conditioned media from reactive astrocytes caused death of neurons within a very short number of hours. So we'd see process retraction and blebbing within around 2 to 4 hours, and then actually death of these cells around about 6 to 8 hours onwards. And we've labeled it here with an Ophidium homodimer. And down the bottom here, we've just quantified this. We've got the vertical axis showing the sort of percentage survival of these cells. And with an increasing concentration of the non-toxic or control astrocyte conditioned media, these cells are very viable. With an increasing concentration of the neurotoxic or neuroinflammatory active astrocytes, we see these cells totally annihilated. And if we take this very high dose, we can actually heat and activate or protease treat it and bring this viability back to normal. And so we interpreted this to say, well, there's a protein that is released by these astrocytes that's neurotoxic. And we tested this in a whole host of other neurons as well, retinal ganglion cells, hippocampal neurons, spinal motor neurons, cerebellar neurons, a whole host of them, and they were all equally susceptible. The other thing that we did, because it was difficult for us to get sufficiently high numbers of neurons to these assays, we had shown that oligodendrocytes were similarly responsive to this toxin. So here we have oligodendrocytes in phase that have been treated with non-toxic conditioned media from astrocytes. They're very healthy. Here they are when they're treated with the toxic media. You can see they've retracted those processes and blebbed up. And then again, we look at the percentage for survival with quiescent ACM. They stay alive for a long time. With this reactive ACM, again, like the neurons, we can totally annihilate. So what Kevin then decided to do was we actually took these immunopanned astrocytes. This is a really important step here, because if you grow these astrocytes in serum-free cultures, you actually can't recapitulate these effects because they won't enter this reactive state. So we take these immunopanned astrocytes in serum-free conditions. We treated them with either PBS or this pro-inflammatory reactive astrocyte cocktail. We collected both the cell lysate or the astrocyte conditioned media, which we could precipitate or cut out and do some in-gel digestion, and then essentially do mass spec and say what proteins have changed. And when we do this, we see that there is a number of astrocyte reactive proteins that are upregulated, in both the cells themselves and also in the ACM. And given we knew this was a secreted protein, we guessed it was one of these secreted molecules. And when we look at these molecules, we see a whole host of very similar and common friends that we've seen before, things like C3, which I told you would mark these cells, serpin-1G, TNF activating proteins, things like this. We actually tested all of these proteins and showed that none of them were neurotoxic to these neurons if we kept those neurons in a healthy state. So the next thing that Kevin decided to do was, well, maybe the neurotoxin is very, very lowly abundant, but very, very highly potent. And so we decided to get rid of everything that wasn't the neurotoxic protein so that we could look at very lowly abundant proteins. And so I won't run through all the specifics here, but essentially we used anion exchange columns, and all of these graphs are going to show you the percent survival on the vertical axis with multiple versions of a column on the bottom. And what we're looking for in each instance is the lowest viability. So with anion exchange column, it's the flow through that's toxic. Cation, it's the flow through. If we look at a hydrophobic interaction column, it's the lower concentrations. If we do a salt cut using ammonium sulfate, the proteins precipitate out around 60%. And so we can do all of these sequential purifications to work out exactly what is left. And when we do this, we see a whole host. If we look at the spectral count here from the MS, and this is the reactivity media versus the control media, we can see upregulation in red of a lot of reactive astrocyte markers. Blue is a lot of classical secreted proteins like lipocalin-2 and things like this. But we saw here in gray an upregulation of a whole host of different lipoproteins, which was unexpected. And we actually went back and quantified this. We can see if we look at the total amounts of these proteins, alpha-lipoprotein E was increased by around 60% in the reactive astrocytes, and alpha-lipoprotein J or clustrin was increased around 80 to 90%. So now we have this sort of sequential method. We're doing now size exclusion followed by salt precipitation, for a salt cut, a hydrophobic interaction column followed by anion exchange and the cation exchange column. And if we look now at the survival of these cells, if they're untreated, they're very healthy. If we look at the purified reactive astrocyte conditioned medium, all of this sort of effect of this toxin goes through this sequential purification step. And if we purify astrocyte conditioned media from non-reactive astrocytes to make sure that we're not just enriching for something that is toxic at high levels, we can see that those cells are also viable. So it's very, very specific to these reactive astrocytes. So the next thing that Kevin did was actually decided to, well, let's pull out these lipoparticles and see if it's the lipoparticles themselves that are toxic. And so here again, we have percent survival in the vertical axis, untreated cells, reactive astrocyte condition, treated cells that are dying. We did a control, we just pulled out using an IgG neutralizing antibody, and these cells were still able to die. But if we pulled out alpha-lipoprotein E and J lipoparticles, we could actually see that the media that was left over without those lipoparticles was non-toxic. And we did a whole host of other experiments to show that it actually wasn't the lipoproteins themselves, but very likely what they were carrying. And I'm not going to run through all of those complicated data, but suffice it to say, if we take this reactive astrocyte conditioned media here that is able to kill in this red bar, and we elute it using a lipidex or a lipid binding column, we can find the lipids that bind to the column are what is toxic. The non-lipidaceous elution is non-toxic. And also a really important control that Kevin did was not only tested this with a host of different astrocytes, but also tested it with a non-toxic cell, in this instance a HEC cell, and showed that lipids from another cell type themselves are not toxic. This is the elution. So what we did then was essentially the same experiment that Kevin did for the proteins, we did for lipids. And this was working with Gaurav Chopra's lab and Priya Prakash at Purdue University to do these lipidomics. So the same setup as before, we take these immunocloned astrocytes, again, very important that they're serum-free. We treat them to become reactive or stay quiescent. We collect either the astrocyte conditioned media or the cell pellet, do a modified blind dye extraction to get a whole host of different lipids, metabolites, proteins, and run this MRM profiling to look at exactly what's changing in the cells and what those cells are secreting. And when we do this, we can see that actually if we look at the lipids within the cells in this SPC plot, which is sort of looking at the variance between these two groups, we can see in red the reactive astrocytes have very different lipids inside them than the control cells. And the same is also true of the lipids that are released into the media. And this is a little easy to see. I'm just going to jump forward to this, this heat map here. And if we focus just on the lipids that are in the astrocyte conditioned media, so the secreted lipids here, we can see in the top we have 5 repeats of control or non-reactive cells. In the bottom we have 5 repeats of reactive or neurotoxic cells. And what you'll see is there's a marked upregulation of these long-chain free fatty acids. And these long-chain free fatty acids have a carbon backbone of, say, C16 through C34 and above, no branches, no side chains. They're fully saturated lipids. And so we decided to determine whether or not this was actually the molecule that was driving the toxicity in these neurons. So the first thing we wanted to do is we could, of course, purify this lipid and show it was toxic in a cell dish, in a cell culture. We could buy the lipid and show it was toxic in a cell culture dish. We could label the lipid and show it got into the cells and then drove the death of these neurons. But ultimately, what we wanted to do was validate this in vivo. And so what Kevin did was actually looked at the fatty acid synthesis pathway. And what we're focusing on here is this sort of bent arm on the bottom, which is looking at these saturated fatty acids. And we can see our toxic lipids here, C16, 18, all the way through to the C30s, fully saturated, no branch chains. And here we have all these enzymes, elongation or elongation of very long chain fatty acids. And these are the enzymes required to elongate these lipids. And so we just asked some very simple questions. What cells in the CNS actually express these lipids? And we saw that here, for the saturated fatty acid pathway, it's really elongation 1, 3, and 7. Elongation 3 is not expressed in the brain. Elongation 7 is largely unexpressed by oligodendrocytes. And elongation 1, actually, while lowly expressed by astrocytes, is actually expressed by most cells in the CNS. And we validated this using a whole host of qPCR. We also looked at another elongation here, elongation 4, which is involved in the extra long-chain fatty acids. And it's lowly expressed in the CNS, but we found that these lipids were non-biologically available. And there's another elongation, which is elongation 5, which is very highly expressed by astrocytes, but actually doesn't make these saturated fatty acids. It's involved in branch-chain elongation. So elongation 1 looked like our target here. And luckily for us, an elongation 1 flox mouse had already been generated. And so we could use that to try and validate this back in vivo. And so what Kevin did was took a GFAP Cree mouse, crossed it to an elongation flox. So he had a conditional knockout of this enzyme only in astrocytes. He did both in vitro toxicity assays and lipidomics, as well as some in vivo toxicity assays to determine whether or not these cells were able to be protected. And so the very first example here is just looking at some lipidomics assays. And we can see that in red here, I've just highlighted all the fully saturated lipids that were downregulated. Everything on the left-hand side of this graph is downregulated in elongation 1 knockout reactive astrocytes versus wild-type reactive astrocytes. And importantly, then what he was able to do was actually take these, in this instance, oligodendrocytes, but we did this in neurons as well, and treat them with both in wild-type cells with both reactive and concentrated reactive astrocyte conditioned media. Again, we can see that these cells die on the right-hand side of these quantification graphs. But if Kevin took media from these elongation 1 reactive astrocytes, so they're knocked out for elongation 1, they still become reactive, but they don't produce these neurotoxins. We can see that while there's still some cell death, it's markedly decreased. And this is quantified on the right-hand side here for us, showing that the reactive conditioning media cannot kill these neurons. But if we concentrate it around 75 to 100x, we can see some death of these neurons, suggesting that there are other lipids likely involved or molecules involved in this toxicity as well. But this was in a dish. We wanted to also see this in vivo. And so what Kevin also did was took this elongation 1 conditional knockout mouse and did this in vivo toxicity assay where he was doing an acute optic nerve axotomy. And we'd used this model previously and showed that we could actually block the formation of reactive astrocytes or these neurotoxic reactive astrocytes and preserve neuronal health and function. And so Kevin did this again. Here we can see just marker for this retinal ganglion cell neurons RBPMS. In the control setting, in a wild-type mouse, there's lots of neurons. In the optic nerve crush, there's a decrease in the number of these neurons that's quantified here for us. There's a decrease following the crush. But in our elongation 1 knockout, while there's similar numbers of neurons to start with, there is a decrease in the degeneration of those neurons in our axotomy model. And that's just shown here. While there's still a decrease in numbers, it's markedly better than what we saw with our wild-type neurons. So this is just to sort of summarize and say that, microglia seem to be very able to induce this reactive astrocyte phenotype during inflammation and at least in a number of chronic neurodegenerative disease models that we've looked at. And we know that they can actually release this neurotoxin. And the one that we've been able to identify so far has been this long-chain fatty acid family, everything from C16 or 18 through to C32 or so. We also know that we can actually block the enzyme required for the formation of those long-chain fatty acids. It does stop the formation of those lipids. It does preserve neuronal health largely, not completely, but largely, which is really nice, making it a novel target. And we know that this preserves neuronal numbers, but of course, we don't know if that's actually preserving neuronal viability. We're now doing electrophysiology on these neurons to see whether or not they're still functionally active and able to actually function as neurons should be functioning, or is actually the destruction or the removal of those neurons an important part of the recovery from this particular trauma. So just to summarize very briefly, I talked a lot about this ALS study today. We've also done some similar studies looking at a mouse model of glaucoma and shown very similar results. We know this is not true to one model, it's true across several models. And these seem to recapitulate the functions that we see in human cells. We also know from our work in glaucoma that this toxic lipid doesn't just kill all neurons. These neurons have to be susceptible, stressed, damaged, something like this, to be killed by these long-chain fatty acid lipids. If the neurons are otherwise healthy, these lipids have no effect, which is really interesting. And we know, at least in the glaucoma model, but we don't know in chronic models, that if we stop these neurons from dying, there's a period where they're electrophysiologically aberrant, but then they actually preserve and restore this electrophysiological function outside of that window. So we think this is a worthwhile druggable target to focus on these reactive astrocytes. And I mentioned right at the top of the talk that this response, at least for this one particular subset of reactive astrocytes, is evolutionarily conserved. We've done this in both rodents and a whole host of different models, but we've also done this working with the New York Stem Cell Foundation, looking at human iPSC derived astrocytes as well. Now, I did want to try and squeeze in a few moments, but unless someone tells me I'm completely out of time, I won't do this. I'm looking for someone to say no or yes on the computer screen here. We've also investigated this looking in human AD postmortem tissue. This is a paper that was published in print just yesterday in Neuron. And we've also been investigating this in the APPS1 mouse model of Alzheimer's disease. So I'm looking for whoever I can see. I think it's Rebecca, Dr. Matsas, to say yes or no time-wise, if I should talk for a few extra moments or go straight to questions. She's saying yes. So I apologize to everyone. We're going to quickly run through a very quick rundown of these two new papers, but happy to take lots of questions about them. So this is actually some work that was done by Philip Hazel in the lab and Emily Castranio, who's a postdoc in Michelle Ehrlich's lab at Mount Sinai School of Medicine. What we did, or what they did, was using the APPS1 mouse, they did a whole host of spatial transcriptomics. And so what these UMAP plots are showing us is not actually individual cells, but individual spots from spatial transcriptomics. And we're going to analyze them as if they were cells, and this will become apparent as to why in the next few slides. So we can see we capture, thousands of spots in both wild-type mouse and APP mice, and we can ask what's actually different between these two mouse models and where do these individual clusters of cells or clusters of spots actually reside. And it turns out that transcriptomically we can define 26 different clusters of responses or clusters of cellular responses across these two models. And if we start asking questions about, well, where do these cells sit? If we focus on, say, cluster 7 here, which has a lot of genes that are highly specific to that cluster, that is, they're not connected with genes expressed by another cluster, we can see that they exist in, say, the white matter tracts and subcortically, but they're not really present in the cortex or the hippocampus of these mice, these APPS1 mice. We can also do this for a whole host of other clusters. We can see cluster 2, for instance, which is a very small cluster, not highly represented, is really only present in the deeper layers of the cortex. Cluster 17 over here on the bottom right is restricted to the hippocampus. Cluster 0 here is restricted to, say, thalamus subcortical areas. So we can define exactly where these individual clusters of cells exist by using what we call a module probing approach, where we take hundreds of genes that are differentially expressed by that cluster and then probing back into the spatial transcriptomic data. So one of the things we can also do is say, well, is there anything that's specific to the APP mice compared to the wild type mice? And it turns out, well, yes, there is. There's this one specific cluster, the only cluster that was specific to the Alzheimer's disease mouse that we could not see in the wild type mouse. All of the other clusters we could see represented across both models. However, there was upregulation of a lot of pro-inflammatory, reactive, etc. genes in the APP mouse. This particular cluster was highly specific to the AD model. And we got excited by that because what we could actually do is start probing back into the spatial tissue and say, well, where do they reside? And we called these new pigs because Bart de Strooper a number of years ago had published a beautiful paper in Cell talking about plaque-induced genes, pigs. And these new pigs just simply have a broader array of genes that are differentially expressed. And we can see these hot spots here with the warmer colors showing the upregulation of those genes, largely in the hippocampus, here, and in the cortex. And if we squint, we can suggest that there are some spots there, and we may want to argue that those are plaques, but really the resolution is not good enough. So what Philip did was applied this Bayesian next nearest neighbor mathematical operation that was published by Ed Zhao in Nature Biotechnology last year. And what this does and enable us to increase the signal to noise ratio in our spatial transcriptomic data. And it does this by cutting each spot into 6 subspots. And so what we can see now is this is a module for several hundred genes induced, we're suggesting around plaques, same as on the left here, but it's been applied with this enhancement of resolution. And we can see now we have some individual spots running through the cortex and hippocampus. And so what we can then do is actually overlay this with some 6E10 plaque staining. And we can see, for instance, this beautiful spot here, there's a plaque stain underneath with a perfect new pig module hexagon right at the top. Same is true down here. And so we feel very confident that these spots are indicative of these plaques. Now, the other thing that we can do is we can ask, well, what about other reactive astrocyte subtypes that we've talked about? We can module probe for those as well. So on the left here, we just have our plaque-induced gene module. And on the right-hand side here, we have a module which is indicative of this interferon-responsive reactive astrocyte subtype that Philip had published last year in Nature Neuroscience. We call these DUMAs. And we can see that they also localize to the regions of pathology to these plaques in this mouse. So that's all in rodents, and that paper is available on bioRxiv. But we also have this paper that came out, like I said yesterday, which looked in both humans and rodents, looking at human AD tissue and rodent inflammation-induced tissue. And what we actually do, and this is spearheaded by Michael O'Day, a graduate student at the lab, and Jessica Sadik.
Ones, and are the ones that secrete the neurotoxin, or they are a different population from the GFAP reactive astrocytes?
Yeah, that's a great question, Dimitra, and I apologize, sort of skipped over a few of the intermediate points there. So you're completely correct. So the C3 positive reactive astrocytes, we have only ever seen them to be a very small population. If you were to take the entirety of astrocytes throughout the diseased brain, there may be 1 or 2%, and what is just an artifact of the sequencing as well, which is a really, really, really important point to bring out as well. Sequencing itself can also be very artifactual.
Yeah. Okay, thanks. Thank you very much.
Shane, I would like to ask you a question, actually. I mean, you've very nicely shown that microglia activate astrocytes, okay, and they become neurotoxic.
Mm-hmm.
But in the sequence of events in a neurodegenerative disease, right? Have you got any idea what happens first and what happens then? I mean, there is the neuronal part
right.
that degenerates. And is it that neurons somehow have an inherent propensity to degenerate? They activate microglia activate astrocytes, or perhaps there is a different sort of cellular molecular events?
Yeah, this is a great question, Rebecca. I foolishly changed one of my cartoons. We used to have an old cartoon that had exactly that sort of rotation, that is it the neurons that become sick first, maybe they don't degenerate, but they become sick and they release some factor, some alarmin, maybe they present a receptor that would make them susceptible. That would mount an immune response. The immune response would mount the astrocytic response, and then the astrocytes would interact with the neurons. I actually think that's probably what it is. I think it's not a one-way street. I don't think it's always immune cells, because immune cells are responding to something. They're not just rogue, although I'm sure there is going to be an instance where there is just a rogue immune cell. You know, sometimes bad things happen. Cancer is a perfect example. There's no evolutionary sense for cancer. It happens and it's really bad. And this may be that as well in some instances, but I actually think it's a really important physiological role to have this cycle moving through. And so how would killing a neuron be physiological? That's the big question, right? And the way I look at this is tucked away in the supplemental material from Kevin's paper in Nature last year, is the fact that all cells can take up these lipids. All cells, they can take it up. We can see them in there, but only mature neurons and mature oligodendrocytes die. And then another important piece of information is from Kjell Molgaard's lab in Copenhagen from a few years ago. He stained human embryonic brains with C3, this marker of neurotoxic astrocytes that Demetria just asked about, and saw that in regions of stem cell niches, up in the hem, the antehem, around the ventricles, et cetera, neurogenesis, there's a huge number of these putatively neurotoxic astrocytes present, these C3 positive astrocytes. But we know now that they're actually not killing neuroprecursor cells. We know they're not killing oligodendrocyte precursor cells. But what they do is have this functional alteration that's really important developmentally. They don't form synapses. They don't allow oligodendrocytes to wrap axons, et cetera, which is really important. If you're making new cells and they're migrating away, you don't want them synapsing right where they're born. You want them synapsing when they've extended their processes. So I think that we think of these as bad or neurotoxic or detrimental only in the context of the adult brain, but actually developmentally, they're really important. So if these were discovered in an embryonic brain, we're probably talking about developmental astrocytes, not reactive astrocytes. And when the brain is damaged, it needs to replenish or regenerate. And so what you do, you just incite the machinations that you did when you were developing the brain in the first point. It just so happens that in the context of the adult brain, that could be detrimental. So that makes this a little bit more complicated, right? Because you don't want to just ablate the astrocyte, which is what we had been doing originally. You want to just remove individual functions that are detrimental. And that's why we think removing just the production of the lipid is detrimental, because the mice then seem fine. The neurons seem fine. Everything seems fine. All that we're stopping is them dying, and we're giving them the opportunity to regenerate. You need the astrocytes there to provide the trophic support. You know, neurotoxic astrocytes don't stop producing neurotrophins. They just also produce neurotoxin. And so we just need to modulate their function, not get rid of them entirely. So I like this idea of everything connected, cycling back and forth. The other reason I like that is it means that you can intervene at any point. You can intervene at the microglial level. Lots of people investigate that. You can intervene at the neuronal level and try and make them less susceptible or protect them. And lots of people are doing that. or you can intervene at the astrocytic level, which is what we do. And I think what will happen is collectively we'll have like a co-therapeutic that has multiple approaches that will be beneficial for patients. But yeah, I think we're thinking about it the same way.
Yeah. So you're intervening after the neurons have become sick with the bringing down the lipid pathways. So even there is a reversal of degeneration or neuroprotection.
Yeah. Is there a reversal? I don't think that there's a reversal. We're doing this study at the moment, so I can't say definitively if there's a reversal. There's a bet in my lab. I don't think that there is. I think that what we do is we stop progression, right? We don't stop initiation, and Kevin showed that really nicely in his ALS paper in that. So we don't stop initiation. That is probably a neuronal thing, or it could be a microglia interacting with neuronal thing. And Eric Huang at UCSF had a beautiful paper a number of years ago, also in Nature, showing that in progranulin mutant or progranulin knockout neurons, they can become susceptible to molecules released by microglia that they wouldn't normally be susceptible to. So there's definitely something at play on the neuronal side with susceptibility. We've only looked at genetic mutations on the astrocytic side. So we're missing a big chunk of that puzzle there, which is really important to keep in mind. I don't know if that addresses your question, but it's something that we struggle with a lot. I think it's a really big open question that we need as many people working on as possible. I think it's really, really very important.
Thank you. There is one question in the chat from Vasiliki Papadopoulou. Thank you, Dr. Liddelow, for this presentation. I would like to ask, is there a known mechanism or a hypothesis about how these long-chain fatty acids released from reactive astrocytes kill neurons?
Great question, Vasiliki, and I apologize. I took this out of the talk right at the last minute to throw in the AD stuff. So this is a great question. So in the original paper in 2017, we showed that this was a caspase 2-3 dependent apoptosis. It seemed not to be ferroptosis, necroptosis, glutamatic cytotoxicity, et cetera, really apoptotic. But we couldn't really do much more than that because we didn't know what the molecule was. It was all a bit dirty. In the follow-up paper, the paper from last year that Kevin is the first author on, the lipid paper, he was able to show that it is indeed apoptosis. It's likely lipoapoptosis, which is essentially apoptosis induced by lipids. And the lipids engage the PERK-ATF3 PUMA sort of cell death pathway, and it sort of inserts into all the amazing work that's been done on that pathway, which has been investigated to the nth degree, which is really great. So we can actually knock out, say, PUMA or something like this and actually negate the cell death in a culture dish, which is really, really nice as well. So it goes straight into, like I said, PERK-ATF3, ultimately cleavage of caspase 3. And we sort of did Western blots of every single component of that pathway to show when you treat with non-reactive versus reactive conditioned media, you get the switch with the reactive media in these cells. They essentially switch straight over to the cell death pathway incredibly quickly. Again, that's the thing here. It's incredibly quick. Within 2 or 3 hours, this is engaged and those cells are off down the pathway of cell death. And at that stage, we don't have the ability to cling them back from death, but we can stop them from starting that pathway, which is really important. Yeah.
Pasteur has a question.
Yes, Shane, thank you so much for this amazing talk. Very, very informative and helpful. Actually, my question had to do again with the susceptible neurons to these long fatty acids that produce from the reactive astrocytes. And I was wondering if you have shown this the same effect on the Alzheimer's disease neurons, because you said they have to be susceptible in order, after brain injury or something. So what about neurons from AD?
Yeah. Oh man, I just submitted a grant on this. I think this is a really important question. I think this is really important because there is a multitude of AD-associated GWAS that we don't know what the functional outcome of those is, right? And a number of those genes, around about 20% of them, are expressed in neurons. And there's a whole host of, even outside AD, there's a whole host of disease-associated mutations in genes expressed by neurons. I used progranulin as an example just before an FTD. That does increase the susceptibility of neurons. And so what we want to do, and we work very closely with Jeff Bucher here at the Center for Excellence in Genome Science here at NYU, and we're really interested in working out what these GWAS hits do functionally to cells in the brain. Part of what we started doing was focusing on how do they change the reactivity response in astrocytes, and I mentioned the work today with SOD1, lowering the threshold, but part of that is also what's happening on the susceptibility side on the neurons. I think this is incredibly important because it's not just astrocytes being bad and killing neurons. We know that they have to be susceptible. We focused on damage, pathology, but genetic predisposition is going to be a massive part of this. And so, like I said, I've said numerous times, I think this is going to be a co-therapeutic approach, make the neurons or lower the susceptibility of neurons, plus remove the ability of the astrocytes to secrete a toxin, plus probably inflammatory modulation of the immune cells as well, right? Because we know that, as we said with Rebecca, there's this spiral where the cells are all interacting. And so whatever we can do to hit all nodes of that interaction is going to be good as far as I think and see it.
Thank you. Rebecca, thank you so much. I have only one more question.
Sure.
Say, how did you feel about iPS-derived astrocytes? How much can they recapitulate the adult ones and the disease, and also all these different types of astrocytes that we have both in the disease or in the normal brain?
Yeah, this is a really good point. This is something, again, that I think there's a lot of contention in the field about. And I'm fairly on the side of, if you want to study mechanism or function, it's really important to have a lot of control over the system. And that level of control is very difficult to have in vivo. And so you want to really have a base experiment done in a dish and then validated in vivo, right? What we have found, and the reason we've collaborated over the last number of years with New York Stem Cell, and particularly Valentina Fossati there, is that they have a very high throughput automated system. So they're very reproducible and 100% serum free. And that's the really important bit here. Everything that we do in rodent culture is serum free. Everything that we do with Valentina with human iPSCs is serum free. And if you ask any neurologist what serum on the brain is, they say, oh, that's a stroke. That's really bad. You don't want that. But historically, when we studied CNS cells in a dish, we've added serum because it keeps them alive. What it also unfortunately does is it changes the transcriptome and function of those cells, often irreversibly. And so it actually activates those cells. It makes them highly proliferative, which is not a normal thing for astrocytes to do in the mature adult brain. And so for us, for instance, identifying the lipid toxin, we were unable to do that using serum-containing astrocytes because it altered the way that they responded to stimuli. It altered their sort of function in their transcriptome, so they could no longer produce those lipids. So it's a really, really important thing. The other reason that we like Valentina's model of using iPSCs is actually they get generated in like a mixed organoid style culture. So neurons and oligodendrocytes are also present. And we know from work like from Yo Zhang and Chala Eroglu and people like this that astrocytes obviously don't develop in a vacuum. The other cells are really important for that development. And so with the iPSC work, we develop them together. And then we use this marker CD49F to actively pull out the astrocytes at the end of it to do our monoculture function experiments. Whereas with mice, it's a little easier because we can take primary cells right from the get-go and keep them in serum-free culture.
Thank you so much.
Really important point. Yeah.
Thank you.
Let me just get this point clear. I mean, you develop organoids with astrocytes and oligodendrocytes, and then you somehow immunopan or sort of purify the astrocytes. But then again, I mean, do you use them in an acute phase, or do you plate them whereby they may change again?
Oh, very, very good point, Rebecca. Yeah, so we the way that we do this with Valentina is we produce these mixed cultures or organoids, both work. We purify the astrocytes and replate them. And when we track their transcriptome, if you keep them in serum-free conditions, they have stable gene expression profiles. Really very, very important. There is a little bit of a blip when you do the purification, obviously, a day or two. This also happens with our primary purified cells. But if you maintain them in serum-free conditions, within 5 to 7 days, those cells have reverted back to a baseline expression and function. And that's when we would start doing all of our secondary interactions and perturbations on those cells. It's a really, really good point. Sorry, we glossed over that. And Lillian Babar, Lily, who I mentioned, she published a really lovely STAR methods paper in one of the Cell Press journals about exactly that component of the purification and prepping side. If anyone can't find that paper, feel free to write to me or tweet at me. I can pull it out and send it through to you. But it's a really, really important point. You can't do the functional assays immediately. You need to give the cells time to rest and recover from the process of purification. Yeah.
Thank you. So I have another question in the chat. Thanks from Steffi to everyone. Thanks for your excellent talk. Could eliminating saturated fat from diet would have an impact in mediating astrocyte neurotoxicity? This was a second question, and I have another one that I'll read afterwards.
Cool. Steffi, this is a great question, and I also just removed this slide. I'm so sorry. I showed you the cell, so the lipid production pathway, lipoapoptotic pathway of producing these lipids. I actually cut the bottom of that graph off, which shows dietary lipid insertion into this pathway. And when you insert dietary lipids as a starting point, they're actually not fully saturated, even though you may eat them fully saturated. The fully saturated lipids don't make it to the brain to be a starting point for these long-chain fully saturated lipids. So we think, and at least the lipid literature suggests, that the dietary lipids are not what's playing into this role. It's very likely that they're either starting from a very small chain length and being elongated the whole way, which seems metabolically expensive to me. It's probably more likely that starting from some intermediary long chain, but not very long chain fatty acid, which is then getting added to. So it could be from breakdown of cell membrane, phospholipids. You take the phospholipid off, then you've got these saturated long chain lipids there, and that's very likely where it's starting from. But we don't know. We are fast learning as much lipid biology as we can, but the general consensus in the field is that it's not from dietary lipids. I'm still sticking to my Mediterranean diet, though. I'm sorry. I don't know a perfect answer, Steffi, and I apologize for that. It's a great question.
There is another one from Philippos. Congratulations on your presentation. Do you believe that LCFA secretion from astrocytes could contribute to the glial scar formation by clearing the way out of damaged neurons?
Oh, that's a very good question, Philippos. I don't have an intelligent or sensible answer. It's possible. It's very possible. We are investigating this a little bit at the moment to try and determine if these neurotoxic lipids are present in and around the scar. It's probably not in the, excuse me, it's probably not in the scar itself, but it may be, say, on the necrotic side of that scar that it's trying to corral. Remarkably, there is a number of astrocytes that are maintained in that necrotic lesion. Where nothing else survived, there's like some fibroblasts, some peripheral immune cells, and some astrocytes retained. So I like your hypothesis that maybe what they're doing is clearing that out. We know that the lipids don't kill immune cells, so they would be non-susceptible, which would be important. Don't want to get rid of immune cells. We should test fibroblasts, actually, because we know the fibroblasts persist in that region. And so I would hypothesize they're not susceptible, and that would also fit with your hypothesis. I think that's a really good idea that I don't have a good answer to. I'm sorry about that. Maybe we could do a little bit more spatial transcriptomics and some more in situ for these C3 markers and see where they're actually sitting in and around that lesion. Let me get back to you. I'll ask one of the students. I know they're looking at this, and I can get back to you.
And Savas Christoforidis.
Thank you for the great talk. I have a quick question. Are the levels of the lipids, of the fatty acid lipids, high enough to detect lipid droplets inside the cells, or it's irrelevant to this property?
Great question. don't have a sensible answer for you. Terribly sorry. We don't see an increase in lipid droplets in the reactive cells. We haven't done a full quantification of this, so take that comment with a grain of salt, but it's not so remarkable that you see immediately. What we do know is the lipids are largely not free lipids, right? So they have to be contained within something. And it seems to be, I sort of showed one graph of the ApoE, ApoJ lipoparticle pull down. It seems to be that they're incorporated into these lipoparticles. And this is interesting for two reasons. First is evolutionarily, this is important. In the periphery, immune cells, when they interact with, say, an endotoxin from a bacterial cell infection, they take that endotoxin, they put it into a lipoparticle, and it gets excreted or broken down from the body. And we think that maybe this toxic lipid, the same thing happens. The astrocytes make it or have it for whatever reason, like, oh, that's not a great thing to have, put it into a lipoparticle and excrete it. The problem being in the brain, those lipoparticles are really important trophic delivery vessels as well. So they deliver cholesterol to microglia, lactate glutamate, things like this to neurons, and cholesterol to neurons that they need for all of their cell processes and things. And it just so happens now that you have a Trojan horse that contains both trophic components plus this toxic component readily gobbled up by those cells and taken in. Could it be moved around in other things like lipid droplets? Possibly. We haven't seen it in a culture dish, so this could be an artifact of the culture that they don't get packaged like that. So we'd have to validate that properly in vivo and see exactly what's going on. The annoying thing, and this is almost back to Dimitri's question, is up until now, there has not been a reactive astrocyte reporter mouse. You cannot purify just reactive astrocytes from a mouse, which is absurd, right? People have been studying this for decades and that doesn't exist. We've been producing that, so we'll hopefully have it to share for everyone very shortly. And that will enable exactly that sort of question to be investigated, because you could take non-reactive and reactive astrocytes or subtypes of reactive astrocytes and say, what is actually happening in vivo to these cells, just to check if what's happening in dish is different. And I think that's a tool that the field has desperately needed and no one's produced for whatever reason. So we should have that very shortly in a number of months to share with everyone.
Can I ask a final question, perhaps? When you induce these neurotoxic astrocytes, do you see any morphological changes or not only in their basic morphology, but do you see also any vacuoles or anything like that that would suggest other major dysregulation of biological processes?
Yeah, that's a really, really good question. I also skipped all of that. So they definitely do change morphology. So they retract processes, they look hypertrophic, they lose all of their fine, delicate processes. A paper from Mike Sofnu and Baljit Kak a number of years ago suggested these quaternary processes are called leaflets and things. So they do retract all of those. They look a lot more angry, for want of a better term. We didn't look at vacuoles and things like that inside, or didn't quantify, I should say, vacuoles and things inside. The largest difference that we saw, in addition to process retraction, was a relocalization of aquaporin 4 water channels. So normally we saw these only on the end feet, which would of course be wrapping around blood vessels, things like this, makes sense, lymphatic drainage, et cetera. But when they enter this neurotoxic state, we don't know about other states, but when they enter this neurotoxic state, the aquaporin 4 immunoreactivity is also found everywhere around the cell body. And we don't know what that means functionally. There is more, presumably protein there. It looks like more, but it's a little difficult to quantify. We need to actually do this properly, but it is everywhere covering the cell body, whereas before there was none on the cell body and only on those terminal processes. That suggests a remarkable change in function for water homeostasis, trafficking, stuff like this, but we don't know what that is yet. If someone would like to investigate that, that'd be really cool. Come and join us. I just don't know what that means, but it was a really profound change that we saw.
Fine. Thank you very much for this exciting talk and stimulating talk. Basso, would you like to close?
Yes, thank you so much, Shane, for this, as Rebecca said, exciting, amazing talk. We learned a lot, actually. I hope all the students that attend the seminar also feel the same. And thank you all for attending the seminar. Thank you.
Thank you, Shane.
Thank you very much.
I'll come back to you so that you can come in Greece.
Yes.
That sounds fantastic.
Next time we will meet in Greece. Absolutely.
We'll
Bye,
do
all.
Q&A outside. Take care, everyone.
Thank you so much. Thanks.
Thank you. Bye-bye, everybody.
Thank you.
Bye, thanks.
Bye.